So Marinka is an assistant professor at Harvard University, associate member of Broad Institute of MIT and Harvard, and faculty at the Harvard Data Science Initiative. She's a computer scientist studying applied machine learning with a focus on challenges brought forward by data in science, medicine, and health. Her methods have opened up new avenues for understanding nature, analyzing health, and developing drugs, and are used by major institutions. Before Harvard, Marinka was a postdoctoral fellow in computer science at Stanford, and before that, she received her PhD in computer science from University of Ljubljana. While she also researched at Imperial College London, University of Toronto, Baylor College of Medicine, and Stanford at that time. She was named a rising star in electrical engineering and computer science by MIT, and the next generation in biomedicine by the Broad Institute of Harvard and MIT, thus being the only young scientist who received such recognition in both electrical engineering, computer science, and science fields. So, also as many top scientists, Marinka already presented many years ago also at the Amy conference, right, Marinka?
Yes, correct. I remember that conference. I think it was in Pavia. Yeah, it was a very nice experience there.
Okay, so please, Marinka, the floor is yours.
Thank you, Gregor. Thank you so much for these kind words and nice introduction. And thanks to the organizers for organizing this event. I'm delighted to be here with you today and talk about actionable machine learning for drug discovery and development. I hope that many who might not necessarily work at in drug discovery and development problems would also appreciate and find some of the topics exciting and invigorating. Code, data, and papers that I'll be mentioning throughout the talk are all available from my lab website, which is listed here on this slide, zitniklab.hms.harvard.edu, where you can find more information about the material I'll be presenting today. So with that, let's start with an introduction. So what makes biomedical data so unique in the sense that it really requires careful consideration from a machine learning and AI scientist that goes beyond simply applying machine learning algorithms to biomedical data. So over the next few slides, I'll try to provide some perspective on that, and then we'll go into details regarding a few projects. So there are several different aspects. One of them is certainly that for most of biomedical phenomena, there is not a single data set that can capture all the complexity describing that phenomena, which means that we need to integrate heterogeneous confounded data that span from molecular levels to the society. And that data often does not fit into one large flat data matrix, which is a predominant kind of data representation for machine learning models. We need to consider data typically that are multi-scale, span from molecules to individuals all the way to populations, that are heterogeneous in the form of experimental readouts, curated annotations, or self-reported data, and also confounded in various ways as they might come from different technologies and originate from different measurement platforms. Secondly, and second point is that, and which I think it's even more exciting and interesting, is the notion that we need to find ways to translate predictions into actionable hypotheses that can lend themselves directly to downstream experiments, whether those are in wet lab biological research or in clinical settings. And that creates some new challenges. And where do those challenges originate? I would argue that they primarily originate from the fact that, that medical and biological research is primarily about high stakes decision making, meaning that at some point, a human expert needs to make a decision whether that's about which hypothesis predictions to take for downstream experimentation in wet lab biology, or what kind of treatment to recommend to a patient. And those high stakes decisions that human experts need to make really can affect the well-being of individuals. Because of that, we need robust algorithms, and we need to ensure that machine learning models behave responsibly and are trustworthy, that we have checks and balances that are built into ML deployment. Biomedical settings is not the only area where that's incredibly important. This also rises in other areas such as autonomous driving, for example. But as a consequence, that means that having models that simply achieve high accuracy or some perform incredibly well according to some measure of performance, such as area in the ROC curve, precision recall curve, and so on, is no longer enough. So there are other criteria that are incredibly important for these models to be practically useful and also comply with certain regulatory constraints, which means that including explainable predictions and interpretable models that are fair, non-discriminatory, privacy preserving, and so on. And so this gives rise to this broad area of research in machine learning that nowadays is known as trustworthy machine learning. Another and final point that we will touch today that I think also makes biomedical data very unique is that for particular problems that are at frontier of research and frontier of science and medicine, they are typically problems that have very little label data. And so very little means very little in comparison to the size of the label data sets that were used for recent breakthroughs in AI. Recent breakthroughs in AI primarily dependent on lots of label data, and one kind of prominent example that many on the call are familiar with is certainly the ImageNet dataset, which has led to revolution in computer vision and deep learning back in 2000, starting in 2012. And that was really enabled by having this million size image data set where images were labeled. And so these large diverse data sets plus large models have led to broad generalization and success of AI across areas. However, in biomedical settings, that is often not the case, and primarily I would argue for those most exciting problems, then the amount of label data might be quite small, and often our data has really long tail, where for a lot of phenomena at frontier of research, the label data are not available, including modeling of rare diseases, novel drugs in development that I will be mentioning today. So my plan for today's lecture is the following. I will touch 3 topics. So the first topic I'll cover relates to drug repurposing and drug combination and is enabled by new algorithms for learning over knowledge graphs. The second topic will be about finding cures for novel emerging pathogens and diseases never seen before. And the algorithm innovation here will relate to few-shot learning and explainable AI. And then finally, I will mention therapeutics data commons, our new initiative with AI-ready data sets for drug development and discovery. And so with that, let's start with the first topic, that of repurposing and drug combination. And so what is drug repurposing? Drug repurposing or drug repositioning, as known in the literature, is an approach that we are considering as a way to address challenges with development of drugs and therapeutics, in particular, development of a completely new drug from scratch takes on average over 10 years. And costs $2 to $3 billion. And that's really not feasible and extremely resource intensive. So an enticing alternative is then to look at what are drugs that are already on the market or in the later stages of clinical development and see if it is possible to expand their indications and find new tricks for drugs that we already have. And that is exciting because it can really accelerate drug development process, going from 12 to 16 years on average to 6 years and an order of magnitude lower costs are associated with the drugs that can be repurposed for novel, for new uses and diseases. So the shorter timescale is incredibly appealing. And so because of that, the drug repurposing has been quite an exciting research area for machine learning and AI field as well. At the core of this, the core of drug repurposing is the question of what drug treats what disease. So the way to think about this problem, think of it in the following manner. Imagine we have a very large bipartite graph where on the left side we have all drugs that are currently on the market. And on the right side, we have a large number, tens of thousands of different common as well as rare diseases. And so these black links or edges between drug diseases indicate known treatment relationships or known indications. So the goal of drug repurposing is then to identify and predict new edges between drugs and diseases. So the goal is really to design a model that can predict what diseases a particular new molecule or that is in the development pipeline, perhaps in early stages of clinical or preclinical research, or what drug that is already on market, what additional diseases that drug might be useful for and treat. So how to model this problem? You might have already noticed that I was kind of mentioning this notion of graphs in the previous slide, and that is the particular paradigm that we are approaching and using in much of our research. And why that is important? That's important because the way drugs affect human body is not limited directly to the molecules and proteins they target, but those effects really then propagate through underlying biological networks. And so because of that, it's really important to model the interconnectedness or take the network-based approach. We're trying to identify what kind of therapeutic opportunities drug might represent. That goes well beyond drug development as networks or graphs pervade biomedical data across multiple scales. That I will be mentioning today in the talk, going from this molecular scale of proteins that the drug targets and the way those proteins interact with each other, all the way to the level of what diseases coexist in individual, and then to the societal population level in terms of healthcare systems. Our key insight for us in this drug repurposing project here is the notion that we will think of drugs and diseases in a slightly less naive way. What that means is that we will model drugs and diseases not as individual nodes in a graph in graphs, but really as rich subgraphs. So the way to think of it is that we'll start with the human protein interactome. That's a network where nodes here indicated by white circles are human proteins that are connected by edges that indicate physical contacts between proteins. So that's the protein interactome. And on top of that network, we overlay a large number, tens of thousands of different subgraphs. Each subgraph represents a drug and disease and is defined by a rich protein network on disease-associated proteins or drug targets. In pharmacological sciences, it is known that a drug likely treats a disease if it's close to the disease in pharmacological space. So these are very big words and an exciting way to conceptually think about treatment. However, it's not practically useful. How can we operationalize this concept of closeness in pharmacological space? So the idea here is that we use the paradigm of deep graph neural embeddings to operationalize this concept of closeness in pharmacological space. So we will define our problem formulation as follows. So slightly more technically, our goal is to generate for each and every subgraph that we have, generate an embedding such that the likelihood of preserving subgraph topology is maximized in the embedding space. In other words, what that means that if we think of our input as consisting of this rich graph representation of human interactome and overlaid with many subgraphs, we want to map, sorry for the typo, we want to map subgraphs to these d-dimensional embedding vectors such that two subgraphs with similar topology in the input space, in the input graph, two subgraphs that are close in the input graphs, in the input space will be embedded close together in the embedding space. So in other words, that means that we want to specify some nonlinear deep transformation function, which we denote here F. F will map each and every subgraph that we have here, S1, S2, S3, S4, and 5 on this cartoon. It will take that subgraph, it will map it to a point in the d-dimensional embedding space. That point is associated with the vector. This vector is embedding of the subgraph, such that if we then perform linear algebraic operations with those embedding vectors, that reflects topology of the input graph. And so that directly then goes back to this notion of closeness in pharmacological space, because we can replace it with similarity or closeness of embedding vectors between drug and diseases. So details of this approach are described in the NeurIPS paper that we published last year, that puts forward a model known as subgraph neural networks. It's an approach that does exactly that what was discussed in the previous slide. So it's an approach that takes as input as a large number of subgraphs, S1, S2 up to Sn. It specifies a neural message passing architecture, and that architecture then allows us to generate on the fly an embedding for any subgraph of a network. And that those subgraphs, when those subgraph embeddings were obtained, can be then used for treatment prediction. And so how is that done? The high level, the setup is the following. So our input consists now of this bipartite drug disease graph, but actually then each disease is a subgraph and each drug is a subgraph. And these black edges indicate treatment relations. The approach is to take that data as input, learn embeddings of disease and drug subgraphs, and then use those embeddings, measure the distance between them in the embedding space, to predict whether a drug should be indicated or contraindicated for a disease. So that then allows us to predict the new edges between, or new relationships between diseases and drugs. So we did a variety of validation studies for this particular approach. One that I will mention was a collaboration with Stanford Medicine Spark Translational Research Program, which is an international program that has engaged in repurposing studies for the last several decades. So they shared with us a comprehensive list of drugs and diseases that were repurposed in that program, which I'm showing here on this slide. So let's look just at one example. The fourth row from the bottom is an example that indicates that carvedilol was repurposed for Chagas disease. So we then asked, well, what is the rank at which the subgraph neural network model would predict carvedilol as a repurposing candidate for Chagas disease. And so without that data being included or seen by the model at all, the model ranked carvedilol as 9th most highly ranked candidate for Chagas disease in a system of 5,000 of drugs that we were considering in this study. Even if you look at other examples of, you can see that correct drugs were mostly ranked very highly, very close to the top of the ranked list of drugs for a particular disease. And that was particularly exciting and has motivated us to then engage in follow-up studies that we currently have on prostate cancer, where we are looking at a personalized drug repurposing prediction in the context of minimizing incontinence as unwanted side effect. And so that brings me to another topic that I would like to touch upon, which goes away from modeling efficacy, meaning what drug will treat a disease, but it really also considers a second important endpoint, which relates to safety. So really a drug that can be given to patients, not only it needs to be efficacious to achieve its desired therapeutic effect, but it needs to be safe. And so the particular safe for patients that take it. So the particular question that we were interested in this context relates to polytherapy. And so this is the notion when patients take multiple drugs at the same time to treat complex coexisting diseases. It's very common. So the statistics that I have here for the United States is that 46% of people over 65 years take more than 5 prescription drugs. It's not uncommon to encounter patients that take up to 20 drugs at the same time to treat heart diseases, depression, cancer. And the problem with that is that patients that take multiple drugs at the same time are at much higher risk of unwanted adverse events. And so over 50% of the US population are affected by them, which is a problem for individuals as well as healthcare system because of additional costs as well as increased morbidities and mortality. So what is the main issue behind taking multiple drugs at the same time and increased risks of adverse events and safety, drug safety considerations? The key issue is that of unexpected interactions between drugs. So let me illustrate that through an example. So imagine we have a system where we have just 2 drugs, a blue one and a red one. So an unexpected drug interaction refers to the following setting. When a patient takes a blue pill alone, desired therapeutic effects are achieved, no side effects occur. When a patient takes red pill alone, the desired therapeutic effects are achieved and no side effects occur. However, when a patient takes red and blue pill together, then with some probability, unwanted, unexpected adverse events happen. And so that is unexpected because we would not expect to get these adverse events if red and blue drug would act independently of each other. So there are some interactions between those 2 drugs that occur when patients taking them jointly. So our task here is to model and how likely it is that a particular combination of drugs will lead to a particular type of side effect. And so the way we approach that is by constructing a large knowledge network that we call Polypharmacy network. This is a network with that contains information based on our collaboration with the FDA in the US, and that allowed us to pull together a rich data set of adverse events reported by the entire population in the US, which we then encode in the following manner in the knowledge graph. So we represent every drug as a unique node, denoted here by green triangles. 2 drugs are connected by an edge. That edge has a particular type, R1, R2, R3, and those types correspond to side effects. So 2 drugs are connected by an edge if and only if they lead to a side effect that is seen in patients much more often than expected by chance, given the underlying information about this distributional nature of how often the drugs are prescribed to certain populations. And so that means that drugs or green triangles in this network connected by edges are those drugs that in human populations lead to unexpected drug interactions and unexpected side effects. In addition to this mode of information that contains information on interactions between drugs, we encode into the knowledge network information about what proteins are targeted by the drug. And so that then gives us information about how drugs actually work in the sense of their mechanism of action, and then how those proteins interact with each other in the underlying protein interaction network, which gives us this knowledge network altogether that consists of drugs and proteins and over 1,000 different types of edges, encoding different kinds of side effects, and then several tens of millions of actual edges and data points that are in the knowledge graph. So we have recently developed a model that we call NDecagon that can take that polypharmacy knowledge network as input, and it defines a graph neural network architecture. So that is a very powerful a model that I will not describe in details in this talk, but I very much encourage people to check our websites to learn more about graph neural networks. But the NDecagon can be used to get predictions about unexpected adverse events for combinations of drugs. So endpoint users and human experts can ask queries that are of the form, can be of the form, for example, how likely will a form of statin and ciprofloxacin when taken together lead to breakdown of muscle tissue. And in the back end, NDecagon really uses its graph neural network to return the probability of an edge breakdown of muscle tissue with that type existing between the two drug nodes in the knowledge network. So we again did a variety of validation studies. I will just highlight two of those. So in one study, we first asked our model what is its ability to make useful predictions on data that was not seen and available to the model. So to that end, we trained NDecagon on all data generated prior to 2012 and asked the model to make predictions that the model was most confident about. And here in this slide, I'm showing top 10 predictions made by the model. Each prediction comes in the form of a pair of drugs and a side effect that is predicted to be associated with that pair of drugs and cannot be attributed to either drug alone in combination. And then we ask how many of those predictions were confirmed after 2012. And for many top predictions, we're able to find direct evidence in literature in the form of patient case reports that exactly match with predictions made by the model in terms of unexpected adverse events and particular drugs that were involved in that are associated with that adverse event. There were a variety of novel predictions that we were eager to make sense of. And so for that, we have recently engaged in a larger clinical validation of predicted polypharmacy side effects with several hospitals in the West and East Coast, where we are looking where we are using classic drug interaction markers as well as lab values and many other surrogate measures extracted from electronic health record data in order to validate and make sense of some of predictions that NDecagon has made. Now, though, if you paid attention to what I was describing over the last few slides, you might have got a question about, well, does that only work for pairs of drugs, right? Because predictions that I showed on the previous slide were all about a pair of drugs and a side effect. And I motivated this project as there are many patients that take way more than just 2 drugs. So that's a great question. So in the last year, we have worked quite extensively on NDecagon to make it useful for the design and study of ultra high order drug cocktails. And what I mean by that is to model really combinations of drugs that consist not only of 2 drugs, but of many more drugs, up to 10, which is what we are currently able to do. And that's incredibly important and to have such computational support, because the number of combinations of drugs grows exponentially fast. Meaning that it is not possible to simply screen all combinations of a certain order in brute force manner through experimental work. Instead, we need then computational methods that can take as input a large number of drugs and make predictions about their safety. The challenge for that, from the computational standpoint, is that there is very little labeled data available about higher order combinations of drugs. And so the question then is, can we really use information on just pairs of drugs and make predictions for higher order combinations of drugs that consist of more than just 2 drugs? And the way to do that is through an approach that we have recently developed and are currently validating them on clinical studies, which we call NDecagon. NDecagon is an approach that it operates on large knowledge graphs, and what it does, its key idea is that it takes classic operators developed in pharmacology for drug interactions, They are often known under the name of Bliss interaction, Bliss operators. And we came up with the differentiable counterparts of those Bliss models. Those differentiable counterparts now allow us to calculate gradients and propagate them through our graph neural network architecture, which essentially means that we can now train deep models because we have these differentiable counterparts of Bliss models, and that those deep models directly are infused by domain knowledge that originates from our pharmacology, because the architecture of the model itself is inspired by prior domain knowledge in pharmacology. And the appealing part of NDecagon is that it can take as input a set of drugs, no matter what is the size of that set. And that means that we can then, when we train those Bliss operators on pairs of drugs, once they are trained, we can use them as a query answering system that can answer queries about safety of combination of any size. So a piece of 2 points of results about NDecagon are as follows. So we've been working with FDA quite closely, and to this end, we have explored the data set of that contained over 10 million adverse event reports. Those adverse event reports are reports submitted by healthcare professionals to FDA and contain information about individual patients, what drugs they are currently being prescribed, and then what adverse events they are reporting to experience. And so there are over 19,000 distinct adverse events that are seen in that database. And we use information about from that resource in order to train NDecagon model that can then predict for combinations of any size. We are looking at size of 2 drugs, 3 up to 10 drugs, and predict what kind of adverse event that combination might lead to. So this heat map on the left, what it shows performance measured as a recall at top 100, meaning how many hits about correctly identified adverse events do we find in our predictions if we look only at top 100 most predictions that the model is most confident about. So what we can see is that, and this is just a small part of results, we can see that the model really is able to, with quite surprisingly high accuracy, then also predict safety events of combinations of higher orders. Even more interestingly, for the interpretability standpoint, is this UMAP plot on the right that shows that the underlying embedding space that we learned in NDecagon is really biologically meaningful, because it puts together it captures the compositional nature of combinations of drugs, where, for example, we see in purple points drug combinations consisting of 2 drugs that then are combined by the model itself in order to understand the effect of combinations consisting of 3 drugs, shown in this plot as blue triangles, that are then combined together into combinations of 4 drugs, shown as green squares, and so on. And so it's been really illuminating for our collaborators to look at these embedding spaces. And very excitingly, we see that the performance of NDecagon model for drug combinations of varying size is persistent, in the sense that even when looking at combinations that consist of 6 and 7 drugs, we see that performance of NDecagon, shown here as red bars, is is high and considerably better than various simplifications of NDecagon, where we either do not use graph neural networks approach, or we are not using the notion of Bliss operators, which indicates that it's really important to be able to infuse domain knowledge that has been developed over several decades in the area of research, in our case pharmacology, and infuse it into very powerful state-of-the-art machine learning models, in our case graph neural networks, to achieve great performance. Okay, so that is the first, that concludes the first large part of today's talk. Next, I will focus briefly on the second topic that also relates closely to the topic of this workshop today, and that is about finding cures for emerging pathogens and the underlying challenges of that. So I would argue that one of key challenges that is facing biological discovery today is the challenge that an observation that generalizing to new phenomena is particularly hard for machine learning models, because prevailing methods require abundant label information, but really labeled examples are scarce. So that includes, for example, problems that require learning about novel drugs in development. Because drugs are novel, there of course are very little label information and poor understanding of their efficacy and safety. For example, for modeling emerging diseases or diseases we have never seen before, modeling rare diseases, which by definition are diseases that affect very small number of patients, and therefore it's not plausible to even expect to have a very large patient data sets for them at any point in the future, or to model hard to diagnose patients. So pictorially, the problem is that what prevailing methods in machine learning assume is that what we have this setup on the left part of the slide, where we have this underlying data set, in our case graphs, that are richly labeled. So most of the nodes in that graph is labeled by blue, red, yellow color, and given this richly labeled data set, one would predict labels of unannotated nodes in that graph. However, in reality, what happens is exactly the opposite. The data set is very poorly labeled, and only a handful of labeled examples are given. So this is really a challenge that we are addressing and are very interested in over the last year or so. And in the core question that underlies it is how to learn and reason about this never before seen phenomena to make sure that our models generalize to new phenomena as they encounter new patients, new diseases, new cell types in other contexts. So one slide of background for those who are not familiar in few-shot learning, I will use an imaging problem as an example. So an answer to this notion of lack of labeled data examples is a paradigm in machine learning known as few-shot learning. Formally, few-shot learning is an instantiation of meta-learning in the field of supervised learning. But the way to think about it conceptually is in the following manner. So this is a paradigm that in which learning proceeds in two stages. The first stage is called meta-training phase, which is a phase where a machine learning model is trained on a large number of tasks. Each task is defined such that it's very challenging. So for example, in training task 1, the model would be given images that belong to 3 different classes, but for each class, only 2 examples would be given. And so we would ask the model to train a classifier that can distinguish between images belonging to these 3 classes. In the second training task, the model would be asked to solve equally challenging tasks, but just with a different set of 3 classes. And so that is done across many training tasks during meta-training phase. Why? In the hope that if a model can be trained in such a way to very quickly adapt to a new task with very few labeled examples, then in the test phase, the model would be also able to very quickly adapt to a new problem for which we have very labeled examples. So at test time, imagine we would need to build a duck versus dolphin versus chicken classifier. And so the problem is that we would only have 2 labeled examples, so 2 images of duck, 2 images of dolphins, 2 images of chicken, and how can we learn really a classifier that for this very challenging problem? And few-shot learning makes this possible. In our particular context, the question is how to make predictions where we are not really working with images, but we work with graphs as they are the appropriate approach for understanding the way drugs works and drug treatments are instantiated. So our approach to this solution to this problem is an algorithm that is called GMeta. This GMeta is a general scalable graph meta learning algorithm. We published it at NeurIPS half a year ago, and all the details are in the paper, but the intuition is very similar to what I described before. But now, instead of classifying images into classes, we really want to predict labels for nodes in such a way that during training time, the model can see information about a large number of diseases. Each disease is defined as a separate training task. And during meta test phase, the model needs to reason about new emerging pathogen or emerging disease that has never been seen by the model during training time. And the model can really successfully do that. And that is something that we were able also to theoretically show and motivate that GMeta is really a suitable approach for tackling these very hard few-shot learning problems on graph structure data. I will not go into details here, but I'm very happy to answer questions about more questions about it. What I will focus next is really show an example of how GMeta and few-shot learning algorithms on graph really have enabled us quite prominently to tackle very important problems recently. And so for that, I picked one example, which is very easy to motivate. That is a project that we were engaged in spring of 2020, where between March 23rd and June 15th of last year, we were part of a task force for identifying therapeutic opportunities for novel coronavirus. And during that 3-month period, we were able to pull together data sets, construct knowledge graphs, and then use few-shot learning algorithms to identify candidate therapeutic opportunities for novel coronavirus at that time, and then validate them in the wet lab and later on in human cells. Why is this a particularly suitable setup for a few-shot learning and GMeta? It is because at that point in time, in March 2020, very little information was available about novel coronavirus, and obviously no successful or even candidate treatments were available at that point in time. So it was very hard machine learning challenge because there were no labeled examples that one could simply take a more standard machine learning algorithm to make predictions. And so to this end, the way we proceeded as part of that effort was the following. The first obvious question that arose was, well, how do we represent COVID disease in computational manner? So for us, the way that we did that is by relying on data that was available and produced by Gordon et al. And our colleagues from UCSF, who have expressed 26 of 29 SARS-CoV-2 proteins, and then use mass spectrometry to identify what are human proteins that are attacked by the virus. And so that is that viral disease module, which was a subgraph of 332 human proteins that are attacked by the virus, was for us a computational representation of COVID-19. So that was now a subgraph representing COVID-19 disease that we included in our data set, where we had tens of thousands of other subgraphs representing other diseases, as I have mentioned in the beginning of the talk. And then also for each and every drug, we represented with its own that is on the market, which we represented with each own subgraph of drug targets. And then the questions became how to really map what drug would be likely candidate for COVID-19. And so a bit more concretely, that means that our underlying data representation was a large repurposing knowledge graph that we constructed that had the following components. It consisted of the human protein-protein interaction graph, where nodes are proteins, edges are physical contact between them. All US approved drugs and the proteins they bind to, as well as some off-target effects for those drugs. Each drug represented as a subgraph of the human protein interaction network. Then common diseases and their associated proteins. Again, each disease represented as a subgraph based on its associated proteins. Information about known drug disease treatments for common diseases, and then also this COVID-19 disease module of 332 human proteins. Given that data, the goal was, given many diseases and treatments for each of them, our goal was to predict candidate treatments for this new disease that we never saw before, for which no treatments exist yet. And so that's exactly the setup that few-shot learning is particularly suited for, because it's concerned with this task of novel treatments for a new disease. So we use GMeta to generate the embedding space of COVID-19 repurposing knowledge graph. I'm showing here part of that embedding space where every point is a drug or a disease. One point in the space represents COVID-19, and then looking at what are the drugs in the surrounding part of COVID-19 in the embedding space allowed us to produce a prioritized list of drugs that were predicted candidate opportunities for COVID-19. And first we took those lists and we compared them against drugs that were in clinical trial at that development at that time. And we did that to get a glimpse into how potentially useful those predictions produced by these models could be. And those results were very encouraging in the sense that a graph neural network method based on GMeta performed considerably better than a variety of other alternatives that we were exploring in the task force. And so that has then really motivated us to not only look at what drugs are tested in the clinical trial, as that does not indicate those drugs really work if they are tested in the clinical trial. Because of that, we teamed up with national emerging infectious disease laboratories in the US that works with live viruses, and that laboratory has tested most promising predictions made by the model. They have screened those compounds for efficacy against SARS-CoV-2 virus. They first did that in viral E6 cells, which are African monkey cells, and later on did validation on human cells with top drug candidates. That has revealed 77 drugs that had strong effect and were active over a broad range of concentrations. And from the computational standpoint, this is particularly exciting because those drugs that showed strong effect in terms of inhibiting viral growth were ranked very high, close to the top of the ranked list of predicted drugs. And that was an order of magnitude better hit rate among top 100 drugs than what could be used what could be identified alternatively by brute force screening. Now, from the computational standpoint, not going into details in biology, this project to me was very illuminating from the standpoint that it really operated under compressed timescales. It required that we compressed work that is typically done in years or months in actually span of weeks or sometimes even days. And so what that meant is that domain scientists, in our case, biologists, virologists, and clinical researchers had to engage with machine learning models more directly rather it was than just looking at simple lists of predictions in some static table. So the questions that we were thinking about is, how can we enable scientists that can provide feedback on model development and build trust into ML predictions, and then enable them to ask what if questions and receive predictions that can be interpreted meaningfully. And so for that, to address these issues, we have used an approach which is called GNN Explainer. This is a state-of-the-art algorithm for explaining predictions made by graph neural networks, which published also at NeurIPS in 2019. And what it allowed us to do is to explain machine predictions about candidate drug opportunities. So the key idea is that GNN Explainer can summarize where in the data, where in that repurposing knowledge graph, the model looked for its evidence for evidence for its prediction. And the explainer then finds a small subgraph of the entire knowledge graph that is most influential for the prediction. In what sense? In the sense that if that subgraph and the data that is in the subgraph would be removed and hidden from the model would change its predictions in a dramatic way. And so if that is the case, then that subgraph has seemingly crucial information according to the model for the model to make prediction about a particular drug. And so this idea that I'm described now is formally known as counterfactual reasoning, and GNN Explainer is an approach that can generate and explain predictions made by any graph neural network using this counterfactual reasoning. Practically speaking, that now means that our users of our models can not only ask queries, but ask queries and obtain predictions and explanations for them. So an example, more recently, we looked at disease treatment mechanisms where human experts can ask queries just such as, will a form of statin, will rosuvastatin treat hyperlipidemia? What is the disease treatment mechanism for that? And what the model returns is a small subgraph of the knowledge graph that consists of some subset of genes, some subset of metabolic functional pathways and disease and other diseases that, according to the model, are a crucial piece of information for the model to make its prediction. So I would want to emphasize that that does not mean that's a necessarily biological mechanism of action in the causal sense, but it's a mechanism that allowed the model to make that particular prediction. And so that's incredibly useful for our collaborators now, because what they are doing is they use this additional functionality to query machine learning system models, first test them and get understanding of when do they work well, when do they fail. That allows us to debug the models also in terms of data quality, and then also identify what are hypotheses that are most likely to be confirmed later in more resource-intensive experimental work in wet lab laboratories, and then take those hypotheses and predictions made by the model. In one particular application of GNN Explainer to this drug repurposing effort for COVID-19 that I was describing before, we actually use GNN Explainer to try to make sense of what are those 77 drugs that the model correctly identified as drugs that reduced viral infection. Is there something common to them? So by looking at explanatory subgraphs returned by GNN Explainer, we were able to identify that 76 out of 77 drugs are those that do not directly bind to proteins targeted by the virus. Instead, those drugs rely on network-based actions that cannot really be identified by alternative drug repurposing strategies based on structural biology that use docking mechanisms. And so that has now led to a class of drugs that we refer to as network drugs, and we also expand that quite a bit in the paper that I was listing before. Great. So in the final few minutes that I have, I will essentially just highlight one particular project and initiative that I've been very excited about in the last months. And so this initiative is called Therapeutic Data Commons. It's an initiative I co-founded with several other groups at universities in the US, and Therapeutics Data Commons is available at website tdcommons.ai. So what is it about? It is a first of its kind platform with AI-ready data sets and learning tasks for drug development and discovery. So if you are interested in looking at or working on drug discovery and development related projects, Therapeutics Data Commons is a resource that enables that. In particular, it serves as a meeting point between domain scientists and machine learning scientists in order to promote this interdisciplinary collaborative work and advance and also facilitate algorithmic and scientific innovation in key areas for therapeutics. So I highly suggest or recommend checking the website that we have. Therapeutics Data Commons, so TDC, currently includes over 66 AI-ready data sets. AI-ready data sets for us mean that these data sets are not just dumped data tables that we see in various other repositories, but each data set comes in the form of a meaningful data set split. It's associated with a meaningful definition of a machine learning task, performance metric, and performance evaluation strategies. Those data sets span the entire range of the discovery and development process, and also include a variety of supporting functionality, which we call TDC data functions that implement strategies for evaluating machine learning models under more realistic regimes we see in real-world clinical and biomedical implementations, as well as various data processing helpers, data functions, as well as public leaderboards when people can submit their own models and predictions made by their models on the data set, which are then evaluated by TDC. In addition to this resource, TDC includes an ecosystem of tools, community resources that really can promote and facilitate therapeutics machine learning in this area. So with that, I will conclude my talk for today. Thank you very much for listening to me. We covered these three important topics that I hope have shed some light on key challenges that require algorithmic innovation and development of core new machine learning algorithms, as well as opportunities that drug discovery and development offer for advancing the science and development and accelerating the development of safe and effective medicines. All papers, code, and data, as well as pointers about TDC are available from our lab website, and I'm happy to now also take questions from the audience. Thank you.